Good morning, students. Today we'll start the new chapter, that is biotechnology and its application. As we have discussed in the last class, that biotechnology is the branch of biology which deals about the improvement of biological materials for the various purposes. Now see the different fields where we use biotechnology for the benefits of human beings, like in the field of medicine. Whatever the important medicines available nowadays, that is produced through the biotechnology. Next is agriculture. In agriculture also there is a very big role of biotechnology. Then power industry, environmental protection, food industry, chemical industry. So we will discuss one by one the contribution of biotechnology, application of biotechnology in our day-to-day life. Biotechnology deals with the industrial scale production of biopharmaceuticals, biological using genetically modified microbes, fungi, plants and animals. Applications of biotechnology includes therapeutics, diagnostic, genetically modified crops for agriculture, processed food, bioremediation, waste treatment and energy production. Major research areas of biotechnology to supply best catalysts, that are microbes, pure enzyme, in the form of improved organism with better characteristics. To create optimal condition by genetic engineering for a catalyst to function. Purification of protein, organic compounds by developing downstream technologies. Biotechnological application in agriculture. Increase in the food production can be achieved by agrochemical based agriculture, organic culture, genetically engineered crop based agriculture. Green revolution, the crop production increased by using improved crop varieties, better crop management practices and use of agrochemicals, fertilizers and pesticides. Food supply was tripled but was not enough for growing population. Introduction of genetically modified crops was an alternative to obtain maximum yield by understanding genetics of the crop. Some useful aspects of genetically modified plants in agriculture. For example, improvement in nutritional quality, like vitamin A enriched rice, that is a golden rice. Better nitrogen fixation. Tolerant to abiotic stress like cold, drought, salt, heat. Reduce reliance on chemical pesticides. Pest resistance crops. By developing pest resistance crop, we can minimize the uses of chemicals like pesticides and other which are used in agriculture. Reduce post-harvest losses. Increase efficiency of mineral uses by plants. Prevent exhaustion of fertility of soil. Production of disease resistance crop. If this is possible, then we can minimize the use of chemicals in the agriculture. Micropropagation for biomass energy production. Now see the top 10 genetically modified foods like soya, tomatoes, corn, potatoes, cotton seeds, rice, canola, papaya, peas, and dairy products. Pest resistant plants. Biotechnology in agriculture, pest resistant plants are to be developed which can minimize the uses of the chemicals. Development of pest resistant plants reduce the use of pesticides. One such approach is the introduction of Bt toxin to target plant. For example, Bt cotton, Bt corn, rice, tomato, potato, and soybean. Bt toxin produced by Bacillus thuringiensis due to presence of Bt toxin in gene. Mechanism of Bt toxin and Bt cotton. Bacillus thuringiensis, that is a bacteria. Bt toxin is a protein which kills insects like lepidopterans, tobacco, birdworm, armyworm, etc. Coleopterans, beetles, dipterans, that is flies and mosquitoes. Bt toxin gene express itself protein crystals at particular phase of bacteria. Insecticidal protein. Bt toxin exists as inactive protoxins and on ingestion of it by insects, active form of toxin due to alkaline pH of gut solubilize the crystals. Activated toxin binds to surface of midgut of epithelial cells and create pores, swelling and lysis, death of insect. Bt toxins isolated from bacteria coated by gene cry, different types, incorporated into crop, plant depending on type of crop and target pest. For example, cry 1 AC and cry 2 AB, that is the cotton bollworm, and cry 1 AB, that is a corn borer. See the life cycles of Bt cotton plants. Bt gene is transferred from the bacillus into the corn. European corn borer feeds on corn plants and ingests the protein encoded by the Bt gene. The Bt protein penetrates and collapses the cells lining of the gut and the insect dies. Pest resistance plants. Bt toxin is not toxic to all pests or pests become resistant against Bt toxin. Another approach to develop pest resistance crop against nematodes parasites, that is RNA interference, RNAi. For example, Mallory's gene incognita roots of tobacco. Principle of RNAi is to develop pest resistance, silencing translation of a specific messenger RNA. RNAi takes place in all eukaryotic organisms as a method of cell defense. Silencing of a specific messenger RNA due to complementary double-stranded RNA, a type of RNA molecule to enhance or inhibit translation of messenger RNA, binds to messenger RNA and prevent translation. Methods to genetically modified crop through RNAi. Bacteria used agrobacterium genetically engineered to introduce nematode-specific genes DNA. Nematode-specific gene introduced into host plant, transgenic plant. DNA introduced will produce both sense and antisense RNA in host. Since sense and antisense RNA are complementary to each other, double-stranded RNA initiates RNAi. When nematodes infest the plant, introduction of nematodes genome takes place and presence of DS double-stranded RNA silence a specific messenger RNA of nematode, initiation of RNAi. Parasites will not able to survive in host which express interfering RNA. Transgenic plants resistance and protected from parasite. See the picture of that nematodes. Biotechnological application in medicine. Biotechnology in medicine diagnose and treat different diseases with effective recombinant therapeutic drugs, protect from dangerous disease. Through gene therapy, new and healthy genes can be inserted to replace damaged cells. Target action of drug by through study of genomics. Recombinant therapeutics do not induce unwanted immunological responses as in the case of similar products isolated from non-human source. 30 recombinant therapeutics approved for human use and 12 of these marketed in India. Genetically engineered insulin. Deficiency of insulin causes diabetes, which can be controlled by taking insulin at regular intervals. Traditionally, when human insulin were not available, insulin of animal that is available from slaughtered cattle and pigs. Animal insulin triggers some immune response like allergy and some reaction due to foreign insulin and will not be effective. Bacteria were genetically engineered produce human insulin in large quantities as bacteria can easily grow in culture medium. Genetically engineered insulin first created in the year 1983 by Eli Lilly and American company. Insulin hormone to regulate blood sugar level and administered in patients with diabetes. Mature insulin, two short polypeptide chains, chain A and chain B, which are linked by disulfide bridge. Insulin synthesized as a prohormone in all mammals. A peptide, B peptide and additional C peptide absent in mature insulin as it gets removed during maturation. Using rDNA technology, two DNA sequences corresponding chain A and B of human insulin introduced into plasmid of E. coli produce insulin chains separately. Chain A and B extracted and combined by disulfide bonds, that is human insulin. Maturation of pro-insulin to insulin. Genetic engineering of insulin. See how the production of insulin is diagrammatically shown. Synthetic DNA encoding B chain, gene for B chain, then introduced in bacteria, B chain polypeptide, synthetic DNA encoding A chain, that is also inserted in the plasmid, then transferred into the bacteria, and B and A polypeptide chain is produced, and then that is combined with the disulfide bond and insulin is formed.